Good morning, dear student. In previous lecture, we will discussed about cisplatin. So how this cisplatin, that is anti-cancer drug, it is formed. So in this lecture, we will discuss the mode of action of cisplatin. So how this cisplatin is work. So let us discuss the mode of action of cisplatin. So before discussing the mode of action, you should remember certain points, such as the first point is that the cisplatin is given to a cancer patient intravenously. So you should remember. So the route of administration, administration of cisplatin, it is intravenously as a saline solution, sterile saline solution. So sterile saline solution generally containing the salt of sodium chloride. So you should remember saline, saline, that is the salt containing sodium chloride, salt of NaC. So the route of administration. So there is a different routes of drugs administration, that is inhalation, ingestion, contact with skin and body. So they are administered or given to a cancer patient with the help of veins. So it is called as intravenous administration. The route of administration is intravenous, and it is a sterile solution. It is a sterile saline solution. So it contains the salt of sodium chloride. Second important point, that is due to the high concentration of this chloride ion. So when it enters into the blood, so it remain intact. It remain intact. It remain intact due to high concentration of chloride ion. So you should remember, when you administer the cisplatin with the help of veins, then it remains intact. It does not dissociate due to high concentration high concentration of chloride ions, high concentration of Cl minus ion. So you should remember this point. Next important point is that this neutral molecule. So we know that the cisplatin is your neutral molecules. It does not have any charge. When enters into the cell by different routes, such as diffusion or the active uptake by the cells. So you should remember, that is the neutral molecule, that is your cisplatin. Neutral molecule cisplatin enters into the cell by the process called as diffusion, that is migration of component from higher concentration to lower concentration. It is called diffusion. Or it also takes place by active uptake by the cell, active uptake by the cell. So passive diffusion or active uptake by the cells. So the route of administration, that is intravenous route. Okay, it contains the high concentration of chloride ion, and therefore it remains intact. Then this neutral molecule enters into the cell with the help of diffusion process, or it is also due to the active uptake by the cells. So generally, if you consider this neutral molecule, when enters into the cell, it hydrolyze, that is it undergo hydrolysis reaction. We should remember. So let's discuss. So this is your cisplatin. So if you consider pt nh3 nh3, this is your cl and this is your c. So when enters into the cell, it undergo hydrolysis reaction. So do you know hydrolysis? Hydrolysis means the cleavage of bond by the addition of water. So in hydrolysis reaction of this cisplatin, one of the chlorine ligand is replaced by water molecule. So Pt, so here what do you get? Pt NH3 NH3, this is your Cl, and one of the Cl ligand is replaced by the water molecule OH2. Then if you consider the complex having plus one charge, the complex should have a plus one charge. So uh, in cell, the neutral molecule undergo hydrolysis reaction, and the neutral molecule, so this is your neutral molecule, does not have any charge. Neutral molecule is converted into a charge species, into a charge species. So this is regarding to the important points regarding to the mode of action of cisplatin, that is it is given intravenously. Then if you consider, due to the high concentration of chloride ion, it remains intact, does not dissociate. They enters into the cell by the process that is diffusion. After entering into the cell, they undergo hydrolysis reaction. They undergo hydrolysis reaction, and one of the chlorine ligand is replaced by the water molecules. So let us discuss what is happen in cell. So if you consider a cell consists of different things. So let us discuss. So if you consider the diagram, so let us consider this is your cell. So in this case, this is your pt nh3 cl nh3. So this is your cisplatin. It enters into the cell by the process of diffusion or active uptake by the cell, and this cell consists of different components. So generally the cell consists of different components such as DNA. It also contain RNA, DNA, RNA. It also contain mitochondria, mitochondria, mitochondria. Then it also contain some enzymes such as metallothionein, SH. These are metallothionein, metallothionein. So the cell having different components such as mitochondria, DNA, RNA, and this metallothionein. When this cisplatin enters into the cell, it undergo hydrolysis, and we get here pt nh3. This is your again nh3 cl, and one of the ligand, that is a cl halogen ligand, it is replaced by the water molecule, and this is your a charge species. The neutral molecule is converted into charge species, and this charge species interact with your DNA and RNA. So these positively charged species of platinum complex interact with DNA, RNA, and they are useful for blocking of they are useful in blocking of the action of DNA polymerase. That is, these species, positively charged species, interact with the DNA and it will block the action of DNA polymerase. So, what is this DNA polymerase? The DNA polymerase is an enzyme which is necessary for replication. So, this DNA polymerase, the activity of this DNA polymerase is blocked due to the complex formation between positively charged platinum complex and DNA molecule, and this enzyme is necessary for replication. So replication is nothing but the cell division. So conversion of a single DNA molecule into two DNA molecules, it is called division. Replication is a cell division process, so it will stop this cell division process and reduces the cancer. So this is the action, that is mode of action of cisplatin. If you consider, so in this case, let us consider this DNA molecule, this positively charged species interact with DNA molecules. So let us discuss the interactions of positively charged species of platinum with DNA molecule. So if you consider a DNA molecule, so it contains the DNA molecule contain generally the nitrogen bases. So we know that DNA is a helical structure, so it contains nitrogen bases such as purines and pyrimidines. So it contains purines and pyrimidines are the nitrogen bases. Generally, the purines are adenine and guanine. So before discussing the interaction with DNA, we will discuss the diagram. So let us discuss first the diagram of what is actually present in DNA strand. So if you consider, so DNA chain is there. So if we consider a DNA strand, a single DNA strand, it contains purines and pyrimidines. So if you consider the structure, so let us discuss. So this is your adenine. This is your adenine residue. So you should remember that adenine and guanines are purines. So you should remember first adenine and guanine. Adenine and guanine, they are purines. So they are purines. Purines means these are the nitrogen bases containing a six-membered ring as well as a five-membered ring. So six-membered ring, it is attached to the ring, that is imidazole. So if you consider this structure, so it contains a six-membered ring. Also there is a five-membered ring, and it is a imidazole ring. So it is a purines. So it contain adenine and guanine. So adenine and guanine, so they contain one six-membered and one five-membered ring. So these are the nitrogen bases present in DNA molecules. So adenine and guanine, so they are purines. And if you consider pyrimidines, if you consider the pyrimidines, then they are thymine, cytosine, and uracil. So thymine, cytosine, and uracil. So these three are your pyrimidines. These three are your pyrimidine. So you should remember, whenever you consider a DNA molecule, it is made up by nitrogen bases such as purines and pyrimidines. The purines containing adenine and guanine. Generally, it contains six-membered and five-membered ring. And the pyrimidines, it is thymine, cytosine, uracil. So these are a six-membered ring, nitrogen containing six-membered ring. So you should remember the purines and pyrimidines. So this is your adenine. So this is your adenine. This is your adenine. It will forms a hydrogen bonding with another molecules such as thymine. So let us discuss the interaction. That is, this is your nitrogen, double bond O. This is your CH3 is there. Nitrogen is there. It's not nitrogen. Double bond N is there. Here, double bond O is there. So this nitrogen, double bond O is there. So you consider the nitrogen having double bond O, then it is attached to this nitrogen, and this nitrogen, it is attached to double bond O, double bond N. So this is your hydrogen is there. Okay, and there is hydrogen bonding between this NH, hydrogen bonding. Also there is a hydrogen bonding between this hydrogen of this NH2 and this oxygen, hydrogen bonding. Okay, double bond O. And if you consider, this is your thymine residue. This is your thymine residue. So first is your adenine, means it is your purines, and second is your pyrimidines, that is thymine. So it contain only a six-membered ring, containing nitrogen, thymine, and these molecules are attached with the help of hydrogen bonding. So this is a DNA molecule. If you consider, this is DNA molecule. If you consider another DNA molecule, so it contains guanine and cytosine. So if you consider this nitrogen, it is attached to the further DNA chain. This nitrogen, that is NH2 group, involved in hydrogen bonding. This nitrogen is also involved in hydrogen bonding, and this nitrogen, its valency is 3, so it will forms maximum valency, that is, it will forms a 3 bonds. So the nitrogen which is involved in binding with the platinum complex, positively charged platinum complex, is this nitrogen, that is, we consider this as active site. So this nitrogen, it is not involved in hydrogen bonding, and therefore it is not involved in hydrogen bonding. It is not attached to DNA chain, further DNA chain, and therefore it is free, and therefore the active site, that is the nitrogen which is attached to the platinum complex, is this nitrogen, that is free nitrogen. So it is not involved in hydrogen bonding, it is not attached to further DNA chain, and therefore it is involved in binding with binding with the platinum complex. So you should remember, that is your cisplatin. After hydrolysis, one of the Cl is replaced by the water molecule, and the positively charged species interact with this interact with this active site, that is nitrogen. So one of the water molecule get replaced by this nitrogen, and similarly another Cl ligand which is attached to the platinum, it is also get replaced by the nitrogen present. So if you consider, this is a single strand of DNA. So this is a single strand of DNA. So one nitrogen of this adenine residue, it is bind to this platinum complex, and another nitrogen of a same strand, another nitrogen of either purines or pyrimidine, it interact with your platinum complex, and the other Cl is replaced by the nitrogen atom. That is another free nitrogen atom. It is also binds with your platinum, and the formation of Pt here, your NH3, and both uh, ligand are replaced by the nitrogen. Both ligands are replaced by the nitrogen of this DNA molecule, that is either purines or pyrimidines. So this is the active species. This is the active species of platinum complex and DNA molecule. It will block the action of DNA polymerase, that is the enzyme which is necessary for replication, further cell division, and it is very important in case of the cancer treatment. That is, it blocks the activity of this polymerase enzyme, and it will reduces the replication process, that is the cell division is not continuous, and therefore it will stop the growth of further molecules. And this is the active mode, or this is regarding to the mechanism or the mode of action of platinum. So if you consider uh, two chlorine ligands of cisplatin is replaced by the nitrogen atoms of purines, the nitrogen atoms of purines, that is it contains 6-membered and 5-membered rings, purines and pyrimidines. You should remember what is purines and pyrimidines on adjacent bases on the same strand of DNA, single DNA molecule. And due to the geometry, if you consider, trans molecule does not form 1,2 intrastrand addict. So trans molecule, why trans platinum complex? It is not active anti-cancer drug because it does not form 1,2 intrastrand addict such as cisplatin, and therefore it is not useful for cancer activity. While the cisplatin form 1,2 intrastrand. 1,2 intrastrand means what? It will interact with DNA molecule, that is one of the one of the purines and pyrimidine nitrogen bases of same strand, and therefore it is used as an active anti-cancer drug. So if you consider the cisplatin, uh, no doubt it is given intravenously. So this is regarding to the mode of action of cisplatin. It will interact with DNA molecule, and it will block the action of enzyme polymerase, that is it is involved in the process of replication. However, cisplatin, no doubt it is used as an anti-cancer drug. It is given intravenously, but it having certain disadvantages, that is it is irritant. It is irritant. So you should remember this cisplatin, it is irritant, that is inflammation of veins through which it gives, that is the route of administration, that is it is vein, and inflammation of vein, inflammation of vein, because it is irritant in nature. If you consider, it is, if it is escape from vein, if it is not administered properly, or if it is escape from the vein, it will damage the tissue. So second disadvantages is that it will damage your tissue. So inflammation of vein, tissue damage. And if you consider uh, before and after cisplatin infusion, so before and after cisplatin infusion, the extra fluid are given the extra fluids are given and care is taken to ensure the adequate hydration before both drugs enter and before both drugs entering into the body and after uh, its removal. So you should remember that is it escape from vein, it will damage your tissue before and after of cisplatin. So when the dose is enter your body and after given, the dose is given to your body. So before and after cisplatin infusion, extra fluids are given to protect the failure of kidney. So to protect your kidney, before and after cisplatin is given, infusion of extra fluids are necessary. So these are the some disadvantages of cisplatin. Another side effect is that in initial stage after administration of this cisplatin, there is a vomiting. So you should remember vomiting. Then low blood counts, kidney toxicity or kidney failure, kidney toxicity. Then loss of appetite, loss of appetite, loss of appetite, and loss of hair. That is very important. It is known as autotoxicity. 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 That is the hearing loss. Hearing loss. So these are the some disadvantages. However, these drugs having different disadvantages, but it is most widely used anti-cancer drugs for the treatment of cancer. So we are not very much interested in the disadvantages or harmful effect of cisplatin. We are very much interested what is cisplatin, what are the important uses of this cisplatin. So this is regarding to the mode of action of cisplatin. In next lecture, we will discuss the different derivatives of this cisplatin. Generally, the group, that is the ligand is replaced by the other monodentate bidentate ligands, and these are the derivatives of your cisplatin. So next lecture we will discuss the derivatives of cisplatin, their uses and their activity. Thank you.